Hello and welcome back to our YouTube channel. My name is Maria Matuszewska and today we're going to be talking about post-landing procedures and data analysis. So there are various scenarios for the post-landing procedures. Firstly, it might be impossible for the teams to attend the launching of the Kansats. Fundamentally, it means that every step will be carried out by the competition staff, which is definitely not ideal, but it is indeed manageable. Secondly, we might actually be allowed to participate in the launch, which would especially facilitate the execution of the mission. The first scenario means that very detailed instructions will have to be made for the committee and all needed equipment, including protective gear, will have to be provided by the team. For the sake of the video, let's consider the second scenario. After localizing the satellite, the team members responsible for data analysis will have to put on protective gear. That is a PPE suit, which you can commonly see during the pandemic in hospitals. Nitrile gloves, these are one-use nitrile gloves. A face mask and a headgear, in order to minimize additional external contamination. In the first place, as you can see here, this is one of the prototypes we've already used, but we'll also be using it soon. The inlets of the collecting chambers should be cleaned with an alcohol solution, which in our case is IsoRapid spray and sterile goose pads. So the inlet I'm talking about is this one. This is the additional ring, external ring. And now you have to bear in mind that when working with an alcohol solution, so actually any sort of a liquid, you don't want to put the liquid directly onto the filters as it might affect its permeability. So you want to put the liquid onto sterile goose pads. These usually come in packed like this. This is a very big one, but you can just order smaller ones. You can even order a few in one sterile package. So you're gonna put the IsoRapid spray onto the goose pad, and you very gently want to clean the outer layer of the inlet and the inside of it. After the thin layer of liquid evaporates, the inlets can be sealed with parafilm by simply pressing the material onto the edges of the tunnel. Here is the example of parafilm. It is a very stretchy and self-adhesive material. It is commonly used in microbiology for long-term sample storage. It's very easy to use. Basically, you just put the material onto the edges and press the closing lid, or you basically put the whole thing onto the inlet to seal it. Only now can the whole satellite be cleaned with the same solution and goose pads, and after disinfecting the gloves or changing them, transferred to a sterile plastic Ziploc bag. For the previous tests, we've been using a simple Ziploc bag. You just have to make sure that the plastic is not affected by UVC radiation. It actually can be more porous, therefore increase its permeability, so there would be no aseptic conditions there. You also want to be very cautious when working with UVC light, as it is harmful, and now the aseptically packed Kansat can be safely transported to the ground station analysis room. The Kansat will be placed inside the glove box with the needed equipment already waiting inside in sterile, not permeable containers. The whole structure will be once again cleaned with an alcohol solution, and only then the Kansat will be irradiated with UVC for approximately 20 minutes. Afterwards, the satellite will be opened one layer at a time with UVC irradiation sessions in between until the actual collecting chamber will be reached. Here you can see one of the prototypes. It's much easier to use this one to show you, because for the demonstration I would have to cut this one open. Anyway, you can see the outer chamber here. It will be placed more tightly inside, more securely, but anyway, you're going to have the inner chamber inside. There's the outer HEPA filter here and the ring, and the whole system goes here. So you're going to be opening those step by step until you reach this small chamber. This is the actual collecting chamber. And in between this HEPA and the inner F9 filter, you're gonna have the samples. The chamber will be opened to reveal two filters from which the bacteria will be sampled. Ideally, we'll be using liquid growth medium, but it may also be replaced with a solid nutrient agar plate. If a liquid medium will be used, then the filters will basically be placed within the growth medium and then transferred to the incubator set for 35 degrees for approximately 16 hours, or the filters will be sampled from, and the rest of the filters will go into a fixation buffer solution to enable quantification of bacterial colonies. If you're not going to be using the liquid growth medium, you can also use the sterile nutrient agar plates. Those are very easy to use. You can order them like we did from Thermo Fisher. They come in one pack. You can also have less in a sterile pack, but it's very handy to have them like this. Either way, the probes will be sealed via the parafilm, once again this very stretchy material, and will be transported to the laboratory, or if impossible due to pandemic conditions, will be left within the glove box. The procedures will be almost identical with the nutrient agar plate, so we're not going to cover them now. Some of the microorganisms obtained will be sent to the laboratory, where the team will conduct quantitative and qualitative analysis of the samples, and some will be observed under an optical microscope to describe the bacterial morphology and calculate, for example, the rate of growth, meaning the number of new colonies formed over time. Examining microorganisms under an optical microscope is quite an easy task. Therefore, in this video, we're going to focus primarily on the flow cytometric analysis. First and foremost, let's define what flow cytometry is and the basic principles of operation of flow cytometric analysis. So, flow cytometry is a technology that simultaneously measures and analyzes multiple physical characteristics of single particles. It is usually cells, as they flow in a fluid stream through a beam of light. The properties measured include a particle's relative size, granularity, relative fluorescence intensity, and internal complexity. These characteristics are determined by recording how the cell or particle scatters incident laser light and emits fluorescence. In the flow cytometer, particles are carried to the laser intercept in a fluid stream, and any suspended particle or cell from 0.2 to 150 micrometers in size is suitable for analysis. Any fluorescent molecules present on the particle fluoresce, so we're going to be staining the particles we have with special dyes to basically enable the recognition of emission of light. And now, a fluorescent compound absorbs light energy over a range of wavelengths that is characteristic for that compound. And this absorption of light causes an electron in the fluorescent compound to be raised to a higher energy level, as you can see on the diagram. And now the excited electron quickly decays to its ground state, emitting the excess energy as a photon of light. This transition of energy is called fluorescence. Light scattering occurs when a particle deflects incident laser light. And the extent to which this occurs depends on the physical properties of a particle, usually its size, internal complexity. Other factors that affect light scattering are the cell's membrane, granule material, cell shape, surface topography. All of those contribute to the total light scatter. We're going to have the forward scatter and the side scatter, as you can see on one of the analyses we have performed previously during our tests. Taking into consideration the above, it is crucial to choose proper dyes and buffer solutions for the analysis. For example, during our most recent test, we used the blue fluorescence dye DNA staining called HESHT 34580, which when used on E. coli gave a positive result. We're still developing the methods of cytometric analysis, as we'll be working on various microbial colonies, which basically means that not all the dyes used will work properly, nor will the solutions used. The tests are ongoing and we'll be updating you on the results. Thank you very much for listening. If you have any questions, don't hesitate to comment down below. I'll be more than happy to answer them. You can also email us or just simply wait for the next video. See you soon. Thank you.